When we raised our pre-seed round, one of the biggest challenges was translating a strong scientific and clinical vision into something investors could clearly understand and believe in. So up to that point, a lot of our focus had been on solving a clinical problem and developing the technology. But suddenly the discussion shifts. So it's not just about science anymore, it's about business. So investors want to understand who your customer is, how you will bring the product to the market, what your cost structure looks like, and where the real value lies. What I learned is that in biotech and medtech investment, it's about connecting three elements. So it's strong science, a clear clinical need, and a credible business model.
Translating a regenerative scaffold from concept to clinic raises questions every biotech scientist will recognize. How do you manufacture a biomaterial consistently, define its critical quality attributes, and navigate a regulatory world where devices and biologics rarely overlap? In this second part, Eva-Maria Balet takes us deeper into the process development, clinical realities, and founder lessons behind Regenosca and TissueSpan. There's plenty here that applies well beyond medtech. Let's go back into it. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. On this podcast, we talk about a lot about biologics or cell and gene therapy, and with that, we talk about critical quality attributes. We talk about batch-to-batch consistency and so on. And I'm curious now, as we're talking for a medical device, how does that work in your world?
So I think it's a very good point, and I'm happy that you mentioned this. TissueSpan is a medical device, so we are not creating a living tissue in the lab, but we are creating a mechanical support. So its primary function is mechanical and structural, just to enable the body's natural healing process. And from a regulatory and quality perspective, this actually gives us a strong advantage because we can define and control clear quality attributes. And the key ones for us are sterility, implant dimensions, and mechanical stability. So parameters that are well understood and can be robustly controlled, both during manufacturing, but then also through final product testing. And to ensure batch-to-batch consistency, we rely on a combination of validated manufacturing processes, in-process controls, and final control testing aligned with the regulatory expectations. And because the mechanism is based on defined structural properties rather than biological variability, we can demonstrate a high level of reproducibility and reliability. But just to summarize, while the biology ultimately drives the healing, the product itself is engineered in a way that allows for precise control and consistent quality from a regulatory standpoint.
Let's shift gear a bit, Evie, and let's talk about the progress you've made so far and where you're at and how your clinical studies are going. Tell us a bit where you are right now in the development phases.
So we are very proud because we have treated the first patients. We have developed the technology in the labs of EPFL. We did a lot of preclinical testing in various animal models. And in all the animal models, we could show this perfect tissue regeneration. So showing the regrowth of muscle cells, epithelium, vascular structures, and nerve structures. And based on this excellent preclinical results, we got an approval for a first-in-human study. So we have operated successfully 5 patients. And for us, this is very valuable because like early human results tell you things that no in vitro experiment or animal model can fully predict. So first of all, human safety is key. So you can generate a lot of preclinical data, but ultimately real confidence comes from clinical use. And also many diseases are difficult to fully replicate in animal models. So often you are working in kind of healthy animals, which don't reflect the complexity of diseased human environment. Like this first human study provided insights on safety and efficiency, but I think one of the most important aspects is how the product actually fits into real surgical practice. Can the surgeon use it easily? Does it integrate seamlessly into existing procedures? Does it require additional training? And for TissueSpan, this was very encouraging. So we saw that it integrates naturally into the surgical workflow without requiring major changes. And we could demonstrate a strong safety profile, like with no implant-related adverse events. And importantly, there was a very positive patient outcomes and satisfaction. So it really validates how the product works in the real world.
And what is the indication you're currently focusing on in these clinical studies?
Our first indication is in urogynecology. So more precisely, the indication is called urethral stricture. So it's the narrowing of the urinary tract. And it's a very relevant entry indication because the current gold standard relies on harvesting the patient's own tissue from the inner mouth. So there's a clear unmet need and a strong patient benefit. And in addition, there are alternative treatments for urethral stricture, but they can have failure rates of up to 80%. So there's really a need for better solutions. And what is very encouraging is that based on our first NUM data, we have already been approached by surgeons in other fields. So for example, in prostate cancer surgery, there's interest in using TissueSpan to support tissue repair and prevent leakage, and preparations for a first in-human study are underway. And when you speak about leakage prevention surgery, it's also a major challenge in other areas, such as colorectal surgery, which opens further opportunities. So more broadly, our vision is to make TissueSpan a versatile tool for surgeons, so supporting tissue repair wherever it's needed. And so while we start with a focused indication, we see strong potential to expand into multiple surgical fields and patient populations over time. So our ambition would like to become part of the standard toolkit for soft tissue repair that is available for the surgeons in the operating room.
Yeah, that's excellent. Wow. Are there any limits where you think your current technology will not work, or are these tissue repair mechanisms quite universal in the body?
We are really focusing on soft tissue repair. So I don't really see an application for bone repair, for example. This I would exclude, or like replacing a full hip. We are really soft tissue. So we are speaking about muscle structure, vascular structures, the gastrointestinal space, these kind of structures. For example, when you look from a histological point of view, the urinary tract and the esophagus, for example, they are very similar. They have an epithelium layer inside to protect the below tissue from outside things like urine or food. And under this epithelium, you have a layer of smooth muscle cells that allow movement of the tube.
When I come back to the clinical trials, you mentioned animal models and the limitations. I'm just curious, when you say animal models, is that a combination of living animals and I'd say lab models, in vitro models? What is the percentage between these animal models you typically use in your area?
At the beginning, we used what I call in vitro models. So that means you're in the lab on the bench and you use isolated cells. So this was really keen to check that cells can actually grow our implant, that there's no cytotoxicity. And we were also keen to see if cells are infiltrating our implant or not. So this was all done on the bench in the lab using just isolated cells. But isolated cells are not a living system. So the next step was what we call animal models. So there you really go into a living animal and you operate the animal. And since we already had this indication of urethral stricture in our mind, we have chosen a urethral reconstruction model. So that means you go in an animal and you remove a full length of the urethra, and then you replace this urethral part with an implant and you suture it. So that's actually the first time that you can also really demonstrate that you can suit your implant. And then at different time points, you take out samples to really follow this cell ingrowth, cell infiltration, and also with immunohistochemistry, you can really nicely then stain for different specific tissues. So in our case, it was very important to show that the urethelium, like the inner layer of the urethra, is forming very quickly because this is a layer that it's important to create tightness and to avoid that urine can penetrate into the tissue. And then it was also very important to show that you have smooth muscle cells forming. And it was very nice because at the beginning, you just have like little red dots, which show muscle cells. And then over time, these little red dots, they develop into really muscle muscle bundles. So you can see how the muscle tissue is actually forming. And then when you look, you also discover small circular structures, which are the vascular structures. And that's also important that you have vascularization coming in so that the tissue that is growing is actually also staying alive. We had both in vitro part in the lab working only with cells, and then the animal models where you really go into a living animal and you show the repair mechanism and also the feasibility to handle the implant in a living system.
You have made significant progress already with your technology in your company. As we all know, being an entrepreneur is not always easy. It has a lot of ups and a lot of downs. What was the hardest moment so far in your company history, and what did this teach you?
I think one of the hardest moments for us was around our first in-human trial. So we had just received approval and were ready to start. And then 2 weeks later, COVID hit and everything shut down. So there was no manufacturing, no surgeries. And instead of starting in 2020, we only treated our first patients in 2022. So that was a very frustrating moment because you realize that even the best planning and preparation, there are things completely outside of your control. But what we really learned from that experience was the importance of adaptability. So instead of standing still, we focused what was possible. So continuing development, process optimization, strengthening other parts of the company, and preparing ourselves even better for when things restarted. It taught me that progress doesn't always happen in the way you expect, but it's important to keep moving forward, even if it's in different steps than initially planned.
Yes, I had one director and he always told us, once you made a plan, it's already out of date.
Exactly. You can already put plan B and plan C in the pipeline.
Which is true, but it doesn't replace the planning, doesn't. Another part of being an entrepreneur, and I think where you did make also quite some progress, is on the funding side. You were able to close a pre-seed fund in 2024. So we do know that actually in this period, 2024, 2025, and probably still this year, being able to find some funding is quite a challenge. What was the challenge there, and what perhaps can you also share with the smart biotech scientists how to communicate your science well so that you do get funding?
So when we raised our pre-seed round, one of the biggest challenges was translating a strong scientific and clinical vision into something investors could clearly understand and believe in. So up to that point, a lot of our focus had been on solving a clinical problem and developing the technology. But suddenly the discussion shifts. So it's not just about science anymore, it's about business. So investors want to understand who your customer is, how you will bring the product to the market, what your cost structure looks like, and where the real value lies. So we had to expand our thinking, which indication to enter first, how to build a market, what potential go-to-market strategy looks like, and also what the long-term vision is, including potential exits. And what I learned is that in biotech and medtech investment, it's about connecting three elements. So it's strong science, a clear clinical need, and a credible business model. And you need to communicate that in a very simple and compelling way. So it's like really about storytelling and getting the caption of the investors within the first 30 seconds, more or less. Because in the end, it's not about simplifying the science, but it's about making the value of your innovation understandable and also from business perspective.
I'd like to dive in a bit more in the business perspective because you're currently doing an executive MBA. You are acquiring new business and leadership skills. What are these skills you're building now? And perhaps there's some things you could also recommend a founder actually that they build very early on as possible.
I did this MBA and it was a very good experience. So I finished it in September last year and I did the executive MBA while running the company. And it was actually a very complementary experience. So what made it particularly valuable was that I could directly apply everything I learned to my own company in real time. So from accounting and investor dynamics to communication and strategy. And in a way, I feel like I've been growing together with the company, constantly learning along the way. So the MBA gave me a structured framework and the tools to better navigate areas that are not traditionally part of a scientific background. So especially like finance and business language. And also very important for me gave me the opportunity to exchange with other executives from very different industries. So which really helps to broaden your perspective and to challenge your own thinking. If I reflect on what I would have started earlier, it's probably building that financial and business vocabulary sooner. Because as soon as you step into a leadership role, especially as a CEO, being able to connect the science to the business side becomes absolutely essential.
As we are wrapping up, Evie, what additional question should I have asked?
That's a very good question, because I think you ask a lot of questions. You have touched on our product, you have touched on our science, you have touched on our clinical trial, you have touched on fundraising, you have touched on a lot of things. So if I'm honest, difficult to find another question.
Okay, no worries. With all that what we've discussed today, Evie, what is the most important takeaway from our conversation?
I think one key takeaway is that translating science into real impact requires more than just great ideas. It really depends on collaboration, mindset, and people you surround yourself with. So even this conversation, I think, is a nice example of that. It actually came through networking, through an exchange with one of our collaborating partners who connected us. And in many ways, that reflects how we have built Regenosca as well, by bringing together different expertise and perspectives. So for me, whether it's innovation, entrepreneurship, or opportunities like this, real progress happens through meaningful connections and collaborations.
Fantastic, Evie. Where can people get a hold of you and learn more about your clinical trials and new developments?
The easiest way to reach me is on LinkedIn. So I am always open to conversations, especially around biotech innovation and entrepreneurship. And if you like to learn more about our work, you can also find information on the Regenosca website and the company's LinkedIn page.
Fantastic, Evie. Thank you so much for coming on the show today, for sharing this exciting science. Smart Biotech Scientists, you have all the infos in the show notes. And thank you, Evie, once again for being on the show today.
Thank you very much for the good conversation.
Building something new means solving problems no in vitro model or animal study can predict, and surrounding yourself with the right people before you start. Eva-Maria's candor on the hardest moments, the language of investment, and where this technology could connect is a reminder that mastery is built one challenge at a time. Please leave a review on Apple Podcasts or your favorite platform. It helps other scientists like you discover the show. And thank you for tuning in, and we'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.